Hey guys, this is Professor Martin and this is Medical Microbiology, and we are working our way through Chapter 10, which is controlling microbial growth in the body. So basically drugs. We're talking about antimicrobial chemotherapy. What drugs can we use that will kill pathogens but not harm the host? And we've already developed this selective toxicity, this concept of selective toxicity. It needs to be selectively toxic. Toxic to the pathogen, but not toxic to the host. Everything else about the drugs is gravy, is extra, is nice, but that selective toxicity is the overwhelming drive when we are developing different drugs for this. All right, so we are at the point trying to figure out how these drugs work. We've put them into 5 different categories. So we are going to look at each one of these categories and work our way through. These categories are determined as their mechanism of action. How do these drugs work? What is their mechanism of action? How do they work? So looking through these categories, we're going to evaluate each one of these categories with regards to selective toxicity. Is it good, meaning that it's very likely to harm the pathogen but not harm the host? Or is it poor? We may get a lot of side effects. It could harm the host. Is it a poor selective toxicity or something in the middle? So the first category we're going to look at is bacteria cell walls. What are you thinking for selective toxicity for this category? Selective toxicity against bacteria cell walls. Cell walls are made up of peptidoglycan. Do we have peptidoglycan? No. Do we have cell walls? No. So any drugs that interfere specifically with cell walls, this selective toxicity is very good. We have a lot of drugs in this category because of this factor. All right, so what does it work on? Well, we do have to keep in mind that these are that this category is considered inhibition of cell wall synthesis. So the making of the cell wall, the synthesizing, the making of the cell wall. So I have a bacteria. It has peptidoglycan layer. If it only works, if the drug only interferes with building the cell wall, then it's not going to have any effect on existing cells. Where it is going to have effect is on growing cells. As those bacteria are dividing and growing, they can't build a new cell wall. So this is where these drugs will be effective. So that being said, we need to make sure that we're hitting this bacteria in that logarithmic growth phase. If it's in a stationary phase, they're not growing as much. If it's a cell that has a very long generation time, they're not growing as much. We have to take that into consideration. All right, so how do these drugs work? If we can damage their formation of the cell wall, then I'm basically poking holes in my cell. I'm getting a cell that forms a very weak cell wall. You remember we got those repeated NAG and NAMs, and we have those peptidoglycan linkages, those peptidolinkage. The glycan is the sugar. The peptido part is these proteins that link these sugars. A lot of our drugs inhibit this linkage. So our cell is busy making these long chains, but it can't link them together. Makes for a very weak cell wall. If I have a weak cell wall, water will kill these cells. Here's my cell. It's a weak cell wall. I got holes in it, doesn't hold that. Water rushes in, and I have dead pathogens, dead cells. Okay, so again, I mentioned before, many of these drugs prevent these cross-linkage, these peptide components of the cell wall, and those work through that. All right, as we go through these categories, you'll notice some drugs that are in purple. The textbook has a lot of different drugs in here. I'm going to pull out some for that. So if the drug is written in purple, it's something you should know about. You should know that penicillin, the mechanism of action in penicillin is it disrupts the cell walls. It's in this category. It's not proteins, it's not nucleic acids. Penicillin and cephalosporins, vancomycin, all of these are in the category of disrupting cell wall synthesis. All right, I specifically want to focus on penicillin and other drugs that are in the same category as penicillin because we've kind of seen this before. The drug penicillin and some others, amoxicillin, ampicillin, these are drugs that are considered beta-lactams. What does that mean? If you go down here and if I look at the chemical structure of penicillin, I notice that there is this formation here that they refer to as a lactam ring. All right, this lactam ring is part of the structure for penicillin. There are some bacteria that can inactivate penicillin. They make an enzyme called beta-lactamase. Is this sounding familiar to anybody? Beta-lactamase, blah. Think back to lab. Think back to that bacterial transformation lab. Think back to that P-glow plasmid, and it made that blah gene. And when we put it on plates that had ampicillin, ampicillin is a drug like penicillin that has this beta-lactam ring, the E. coli that were transformed, the E. coli that had picked up that P-glow plasmid, the E. coli that had picked up that P-glow plasmid that was had that BLA gene where it made this enzyme called beta-lactamase that got secreted in the environment, it inactivated ampicillin and allowed those transformed E. coli to grow in the presence of ampicillin. This is what's happening with that, this beta-lactamase. Some bacteria have a virulence factor that allows it to become resistant to penicillins. So penicillin is actually one of the drugs that many bacteria are now resistant to. It's one of the drugs we've been using since the beginning, right? And so a lot of drugs have this ability to produce this enzyme called beta-lactamase that breaks this lactam ring, and then the penicillin is not active anymore. The penicillin does not do its job. The penicillin is not able to kill the bacteria. So now the bacteria that can make beta-lactamase is resistant to penicillin. Okay, so that is the mechanism of action. This is how it works. Do know that penicillin and these other drugs are in this category where they are in this category of prohibiting or inhibiting cell wall synthesis. They're not building cell walls. So penicillin works on that, but if we can inactivate penicillin, then the bacteria is resistant to it. Let's see, I think there's another picture. Again, what does this drug do? This drug prohibits bacteria from making those crosslinks, and that's how you're sensitive, how a bacteria is susceptible or sensitive to that. It can't make good cell walls, and then it just lyses automatically. If the bacteria has the ability to make beta-lactamase, it can now be resistant to this drug. This drug will no longer work. Okay. Okay, so that is the first category. That is cell wall synthesis. And if you look at our chart as we're going through the mechanism of action in regards to selective toxicity, this first category of inhibiting cell wall synthesis, this is good for selective toxicity. Because we do not have cell walls, because we do not have peptidoglycan, any drugs that interfere with that are selectively toxic, harming the pathogen without harming the host. Okay, let's do the next one. I'll do that in another video to keep these shorter for you guys.